It's 8 a.m. So I'll slowly get started here. Hopefully more people will show up. I guess I'll start with announcements. Most of what I've already posted yesterday. I would say most of you probably have read it, but judging by the fact that this part is highlighted, many people had not emailed me back to get the free points. I would assume you have not read the announcement from yesterday. So once again, I'm here pleading you to read the announcements. They're very important. And a lot of them have this little sentence right here where it says, speaking of which, reply to this email and let me know you read the announcement. In this case, for half a point, then it didn't work. Sometimes it's 1 point, sometimes it's more than 1 point, sometimes it's less than 1 point. Point is, it's free points that you guys are missing out. And I've already told you this. So you already know that the announcements are going out there with 3 points. And people are still not reading them. But anyway, the exam will be not this Monday, right? Not in a few days, but a week after that. So there's no excuse. A couple of people said, I didn't know the exam was today. So now you know. You've got a full week to know that that's when the exam is. So next week, we're just going to start the new stuff. We're going to start Mitosis Chapter. That's not going to be on this exam. So you're going to have a full week to prepare for the exam. Today's going to be the exam review. In between now and when we have the exam, if there's some questions you have that I didn't cover in the exam review, then I can cover those. So you'll have a full week to be prepared. So hopefully everybody's will do great and actually do the exam. Speaking of those people who said, I didn't know the exam was today, last time. For those of you who still haven't taken exam 1, you need to take it. You can't take exam 2 until you take an exam 1. So if for some reason you didn't catch the announcement where I posted it and said, all right, here's the retake, contact me and I'll show you where to find it, and you can retake the second or the first exam. And then to remind you, for everybody else, everybody can retake the first exam to keep it fair. So it's just an average of your first score and your second score. So for those people who didn't take it the first time, you have a 0 right now. And if you take it, do the retake and get a 100%, the average of 100% and 0 is 50. So you'll end up with a 50. So for those of you who, had an 80 the first time, and then you retake it and you get a 100, then your average would be a 90. So, it wouldn't hurt you to retake the first exam, even if you got a good grade. Because even if somehow you get a lower grade, I'm not going to lower your grade. You'll just stick with your higher grade. And the retake is open book and not timed and all that. So, any questions about the exams? Okay. Also, just to remind you, this exam on Monday, next Monday, is going to be the same way it was last time. You're going to meet online at 8 o'clock. I'll post it probably a little bit earlier than that, but you can start it as soon as you see it. You have to turn it in at 8:50. You're going to lose 10 points for every minute that you're late. 10 points times 10 minutes is 100 points. So anything after 9 a.m. and that thing's just going to shut off because it doesn't matter what you turn in. You're going to get a 0 if you turn it in after 9 a.m. I said that last time. I'm saying it again. So make sure you know what's going on. And then I know one person who didn't want to take it on Monday, and that's fine. So if you want to take it earlier, that's fine. Contact me and let me know, and we can find a time to meet online. As long as I can see you and see your screen and hear you and know that you're not doing like an open book exam, I don't care how long it takes you to take the exam. So if you want to meet before Monday and have more time to take the exam, you need to contact me soon so we can work that out. Yeah, that's it for exams, I think. Yeah, it is. Any questions about the exams? What else?
No.
I
don't got any questions, but did you say it worked for attendance yet?
No, not yet. Thank you. Good point. I haven't done the attendance for it yet. There's not many people here, so I'm holding off a little bit in hopes that somebody might show up. So yeah, that's it for the exams. What else? Well, I might as well do the attendance word since you brought it up, and it is 8:04. The first word for attendance will be September. September. Since today is the last day of September, send it off, honor it a little bit. Yeah, September is the first word for attendance. And you have to turn those words in by 9 a.m. So email me the words by 9 a.m. If you miss that deadline, you'll just need to wait for me to post the video with the questions and answer those. So yeah, so that's attendance, that's exam, lab. I'll again remind you, next week's lab is online. So we need to meet online Wednesday at noon, unless you hear otherwise from me. Some of you won't have to meet Wednesday at noon online. Because if you go ahead and do the lab and I grade it and you did a great job, I'll just tell you, yeah, there's no need for you to come in. So unless you hear otherwise from me, plan on being there at noon online on Wednesday. And if you don't want to, go ahead and do the lab early if you can. And if it happens to be that you did a good enough job, then I'll tell you, you don't have to come in. Any questions about lab? Okay. Yeah, I guess that's it. Let's jump into it. Before I just go through every single question, because we won't have time for me to hit every single question, or we might. But before I do that, does anybody have specific questions from the study guides you want me to cover? All right, let's just go through it then, one at a time. First of all, it would be helpful to know where to find study guides. So if you go to this classwork tab up top, and then go down here to chapter 5, because that's where we'll start. That's the first chapter that's on the exam. Scroll down here, some more videos. Here we go. Chapter 5 study guide. Now this is shuffled, but luckily for chapter 5, they are numbered. So even though they're not in number or in the correct order, you can write down what number they are if you have problems with it. So number 17, enzymes increase the rate of reaction by doing what? So basically, how do enzymes work? What do they do? Does anybody know the answer to that? Do they bind to the substrate imposter? Do they decrease the activation energy? Do they take energy from the reactants and transfer them to the products? Or do they increase the temperature of the substrates? And to remind you, the substrates are the things that they're working on. No one wants to guess?
Is it me taking the reactant?
That was the most common guess that I saw that wasn't correct. But that's logical. That makes sense. But the answer is, where is it? C, decreasing activation energy. So remember, I'll try to zoom in here if it'll let me. It won't let me. Okay. Anyway, if you remember, enzymes or chemical reactions have that little hump, right? You have that activation energy. Even if there's a lot of energy being put out by a chemical reaction, you need to put some energy into it before it can happen. Sort of like trying to push a rock down a hill, but you have a little bit of a hill to push it up before it can roll down the hill, right? There might be a lot of potential energy in that rock, and there might be a lot of kinetic energy once you get it rolling down the hill. But first, you got to push it over a little bitty low, right? And that's the same situation. That's basically what activation energy is. So yes, energy, enzymes, they speed up chemical reactions or increase the rate of reaction, or even allow chemical reactions to happen by decreasing the activation energy. But I'm glad you guessed, and at least it was logical, so thank you. Any questions with 17? All right, 11, you have a 2 kilogram bottle of water at 0 degrees Celsius. How many calories with a big C are needed to heat up the water to 100 degrees Celsius? And basically, you just need to kind of break this down to what it is. First of all, we have kilogram, right? So that's 1,000 grams. But we also have calorie with a big C, so that's 1,000 calories. So algebraically, those things cancel each other off, right? So you could just think of this as a 2 gram bottle of water, and it could be asking about how many calories with a small c, because again, algebraically, the kilograms and the kilocalories, they cancel each other off. Then let's make it simpler. Let's imagine instead you have a 1 gram bottle, let's say you have 1 gram of water at 0 degrees Celsius. How many calories would it need to take to raise that to 100 degrees Celsius, right? So if you want to raise 1 gram by 100 degrees Celsius, that would be 100 calories, right? Because it's 1 calorie per degree, right? So it takes 1 calorie to raise 1 degree of water by 1 degree Celsius, right? So therefore, it would take 100 calories to raise 1 gram of water by 100 degrees Celsius. And once you figure that out, then you just multiply it by 2, because we're not talking about 1 gram of water, we're talking about 2. So the answer is B, 200 calories. And I would ask if you had questions, but that would take up way too much time. So if you didn't understand that, meet me during office hours. Oh yeah, that reminds me, the other announcement. Office hours will be online today, so I won't physically be in my office. No one usually comes, no one ever comes anyway, so it doesn't matter, but that's fine. But I will be online if you need me. But anyway, back to number 11. I know that's complicated, so if you need help, meet me some other time to explain it to you. Also keep in mind, if there's any questions like that on the exam, there's only going to be one. So if you're feeling overwhelmed, like, oh, I can't get it, don't stress yourself because it's only one question out of about 50. So it's only about 2 points. Don't worry about it. Also, there's probably only going to be about 4 options. So you have a 1 in 4 chance of just guessing the correct answer. And if you miss it, it's only worth about 2 points. Any questions about number 11? All right. Also, obviously, if there's a question like that on the exam, the numbers are going to be different. I'm not going to deal with 2 kilograms and 100 degrees Celsius, right? I'll change it up so it's not exactly the same question that you got on the study guide. Number 32, this is a long-winded question. You can read it on your own. Basically, it's saying some of these organisms have an organelle that expels water, right? So if there's an organism that has something, a way to expel water, that should tell you logically that it must have a problem with water coming in. So then the next part of the question that you should think about is, all right, if it has a problem with water coming in, what causes water to come in, right? So let's look at this. I wish I could draw on this. Maybe I can't. It'd be a lot better if I could. Anyway, if water's coming in, that means the concentration of whatever solute is higher on the inside of the cell than it is on the outside of the cell, right? Because that's how osmosis works. The water flows to wherever the concentration of the solute is higher because it wants to even things out. It wants to dilute it. So if if the solution on the inside of the cell, the higher concentration, that means the inside of the cell would be hypertonic to the outside of the cell, which would be hypotonic. So then the answer is, right there, B. So again, I'm going through these pretty quickly because I want to hopefully knock them all out so you guys can study for the exam. So if there's any questions, either let me know now or ask me later. But if it's a really long explanation, then I'll explain later. So yeah, that's answer number 32. I could reword this to say something like, instead of saying that the environment is hypotonic to the protozoan, again, the protozoan is the particular organism we're talking about. I could say something like, the protozoan is hypertonic to the environment. And I'd be saying the same thing. So keep that in mind, right? Don't memorize these questions verbatim. Just make sure you understand the concepts, right? And the concept here is that water is coming into this organism. Therefore, that must mean it's in a hypotonic environment, or it itself is hypertonic to the environment. Any questions about number 32? All right, 24 is very simple. Substances that plug up an enzyme's active site are enzyme inhibitors. It's that simple. There's not much else to say. I might reword that by saying something like, what do you call something that plugs up an enzyme's active site? Right? But basically, that's the right answer. What am I going to say about this? I should also point out this question's not confusing, but it's misleading, because I just want to point out and remind you that there's 2 types of enzyme inhibitors. One of them do actually plug up the enzyme's active site. And remember, the active site is where the stuff fits into the enzyme. That's where the magic happens, so to speak. And then there's other inhibitors that plug up some other spot that's not the active site, and it changes the shape. So inhibitors is the correct answer, but just keep in mind, inhibitors do other things too, other than just plug up the enzyme's active site. That's a misleading question in a way. So any questions about number 24? All right, number 25 is also very simple. Diffusion is an example of what? And that's probably how I would change it, right? Instead of just keeping it just like that, I would say diffusion is an example of what? Endocytosis? No, because that's when you're bringing in a bunch of big stuff into bringing a lot of stuff into a cell. Passive transport? Yep, that's correct, right? Because you're transporting something and it's passive, meaning it doesn't require energy. So it's just going from an area of high concentration to an area of low concentration, and that's what diffusion is. So that's the correct answer. Phagocytosis, again, that's not incorrect because basically it's a lot like it's a type of endocytosis. So that's just bringing in a bunch of big stuff using vacuoles and stuff like that we've already talked about. And obviously the really wrong answer would be active transport because active transport is the opposite of passive transport. Active transport requires energy, and it's when you're going from an area of low concentration to high concentration. This is a good time to remind you, Some of these questions, I don't know if you noticed on the first exam, some of them are worth more than 1 point. So this question, for example, will probably be worth 2 points. And if you guess passive transport, you will get 2 points, for example. And if you guessed endocytosis or phagocytosis, you would at least get half credit. But if you guessed active transport, you would get 0 credit, because that's the opposite. That's the most wrong. So basically what I'm saying is, just like in exam 1, there's some answers that are so wrong that I make the questions worth more points. And as long as you don't guess the really wrong one, then you got half credit. So also keep that in mind too when you go back and look at your grade. You might want to actually look at the questions you got right and wrong, because you may have gotten a decent grade but actually missed a lot of questions. Anyway, moving forward. Actually, I'm sorry, are there any questions about number 25 before we move forward? Good grief. This whole time, sorry guys, I thought you guys were seeing what we were seeing. You guys must have been like, what is he talking about? But here it is. So now you guys, can someone confirm that you can see the study guide now online?
Yeah, we can see it.
Okay, thank you. And the second word for attendance is silence. But thank you for letting me know. Okay. Next one, number 43. Which environments will be best for the functioning of an animal and plant cell? There's not much to say without really going into this, but that's just one of those things I told you you're supposed to memorize. Now remember, plant cells want water coming in because they have that cell wall. So with water coming in, it keeps the cells rigid, right? And that's what keeps the plant upright. And without that, it becomes flaccid. So plants want water coming in. Animal cells don't want water coming in because they'll explode. And they don't want water leaving because they'll dehydrate. So they want something nice and in the middle. So we'll start with animal cells. Animal cells are the easiest. Animal cells prefer an isotonic environment. So let's see, isotonic environment for animal cells. Okay, that part's correct. And again, we said we want water coming into the plant cell. So there, hypotonic environment, yes, because if the environment is hypotonic for the plant cell, that means the plant cell is hypertonic, meaning the water is going to flow into the plant cell because it wants to dilute at high concentration. So the answer is A. Obviously, if you get this question on the exam, I'll probably reword it. So I might say something like, which environments would be best for the functioning of a plant and animal cell, right? So I might flop it, just something like that. So again, when you're studying these, don't memorize them verbatim. I'm not doing that to trick you. I just don't want, it's easy to memorize stuff. You could give me a study guide for a course I've never taken and give me a week to study it. And I could probably do really good on the multiple choice exam and yet have no idea what happened, right? People can just memorize things and not understand them. Anyway, any questions about number 43? All right, number 44, how are membrane transport proteins like enzymes? Very simply, their growth is specialized to material that they act upon. That probably won't be on the exam, because off the top of my head, I don't even know if I mentioned that in the lecture. I know it's in the book, but you know, enzymes, there's specific enzymes for specific chemical reactions. And when we talked about transport proteins, remember those are the things embedded in the cell membrane that let things go in and out. But then it's not just like a door, which is unfortunately the metaphor that I use. It's like a door opens so things can get in and out. It's a little bit more selective than that. It's like a door at a club that has a bouncer, right? It's like, yes, things can come in and out, but only selective things can come in and out. So any questions about number 44? All right, this is an important question. It's not that hard. Anything that prevents ATP formation will most likely what? And let's not even talk about all the other options, just result in cell death, right? Basically, the important concept that you need to know here is cells need ATP. Without ATP, cells die. So it's a very important concept, very easy question. I might reword it. Just again, the concept that you need to know for number 14 is that cells need ATP to live. As a matter of fact, these past 3 chapters, that's all I've been saying since we started, right? I said, if you don't remember anything from chapter 5, just remember this one thing. Or if you only remember one thing, remember this one thing. Cells need ATP to live, right? So any questions about number 14? Getting irritated, I can't zoom in. I thought there was a way to zoom. Anyway, all right, number 39.
You might be able to put 3 dots.
Oh yeah, thank you. Now we're cooking with fire. Thank you very much. Okay. Blank occurs when a white blood cell goes to the bacterium. That won't be on the exam, but the point here is it's something coming into the cell. So endocytosis is a correct answer. But the fact that we're talking about a white blood cell, a white blood cell engulfing a bacterium, which means it's something solid, then the more correct answer would be phagocytosis, because phagocytosis is a type of endocytosis. It's specifically something solid as opposed to something liquid. So again, that probably almost definitely will be on the exam, but C is a correct answer, D is the most correct answer. Again, if that was on the exam, this would be worth 2 points. And if you guessed endocytosis, you would get 1 point. If you guessed phagocytosis, you would get 2 points. And if you guessed any of these other two, you would get 0 points. Any questions about number 39? When two solutions that differ in solute concentration are placed on either side of a selectively permeable membrane, so picture that U-shaped tube that we talked about when I first introduced you to osmosis, and it had a little membrane in the middle of the tube, and there was little green balls on both sides of the tube, and there was one side had a higher concentration, one side had a lower concentration, so that's what we're talking about. So picture that's the situation, and then osmosis is allowed to take place. The water will do what? This is a very important concept, and I talked about it a lot. Basically, and I'm going to read the options later, but off the top of my head, the answer, in my words, is the water will move to where the solute is a higher concentration, right? The water wants to even things out. So if the water goes into the area that has a higher solute concentration, it's going to, not diffuse it, I'm sorry, it's going to water it down, basically, right? So let's look at the options here. Exhibit a net movement to the side with a higher water concentration. Actually, yeah, that's the correct answer. So that's what I said, just in a different way. So just keep that in mind. Again, if you see that question, it will be worded different. The options will be worded different. I might say something like the correct answer might say the water will move to the side that has a higher solute concentration, which is a lot like what it says there, but not verbatim. Yeah, so any questions about number 30? Okay, the combination of sucrose, sucrase, and water the combination of sucrose, sucrase, and water produce sucrase, glucose, and fructose. Which component of the reaction is the substrate? Here's what we know. We know enzymes are named after the substrate, and we know enzymes are not used up in chemical reactions, and we know enzymes end with ASE at the end. So here's what we can do. Let's figure out what the enzyme is, and then remember that the enzymes are named after their substrate, so then just figure out what the substrate is based on the name of it. So here we go. Sucrase, that ends with an ASE, and not only is it in the reactant side, but it is on the product side. So there we go. We know the enzyme is sucrase because of its name and because of the fact that it's on both sides of the chemical equation. And it's called sucrase, and it is named after its substrate. So which of these words looks like sucrase but is not sucrase? Sucrose. So the answer is sucrose. For the exam, you'll get some chemical reaction, or you'll get this question, and there'll be a bunch of things you probably never heard of, and it doesn't matter. It's the fact that we're talking about sucrase and sucrose and water and fructose and glucose. That doesn't matter. What matters is you can identify what a substrate is and identify what the enzyme is. So does anybody have any questions about number 22? Okay, so again, when you're studying 22, don't memorize that the answer is sucrose. Memorize the fact that the enzyme is on both sides of the equation. The enzyme ends with ASE, and the enzyme is named for the substrate. So you might get this question again with different substances, and also I might not ask you what the substrate is. I might ask you what the enzyme is. Or more than likely, I'll give you two different questions. One's going to ask what the enzyme is, and the other one will ask what the substrate is. Also, in a situation, this question might be worth 2 points. And if you guess sucrose, which is the correct answer, that would be worth 2 points. If you guess sucrase, I don't know, I might give that to you because at least your brain was going in the right direction. And if you guessed any of these other two, you would get 0 points. Any questions about that? Okay. Number 33, if placed in the tap water, an animal cell will undergo lysis, meaning it'll explode. whereas a plant cell will not. What will account for this difference? Well, I've already mentioned it earlier, so there's not much use in talking about it now. Plant cells have cell walls, right? So that keeps them protected from exploding, basically. So the answer is that. Where is it at? There we go. The relative inelasticity and strength of the plant cell wall. Or, in short, the plant cell wall, right? It's the cell wall that's the big difference between the plant cell and the animal cell. Any questions about number 33? All right. Glucose molecules provide energy to power the swimming motion of sperm. In this example, the sperm are changing. Well, it's a molecule, right? So we're going from chemical energy, right? We're starting with chemical energy. And then we're talking about the swimming motion of sperm. So that's kinetic energy. So the answer is going from kinetic energy to, chemical energy to kinetic energy. So A is the correct answer. And of course, these options might change. So instead of it saying chemical energy to kinetic energy, it might say kinetic energy from chemical energy, right? So again, don't memorize these verbatim. Understand the concepts, because I will switch the wording. Not to trick you, just to make sure you're understanding the concept and not memorizing words. Any questions about number 8? All right. Number 1, what molecules are scientists, skip that, it's not going to be on the exam. What compound directly provides energy for cellular work? I kind of said this earlier. I've been saying it all 3 chapters. The answer is ATP. Directly provides energy for cellular work. Yes, we break down sugar to make ATP. Of course, we also break down fat to make ATP, but we didn't talk much about that. But the answer is ATP. Cells need ATP to live. Any questions about number 12? All right, number 6, you're riding your bike and stop pedaling, coasting along the road. Eventually, your bike slows down and stops. So basically, try to picture it. You're riding, and you just stop what you're doing. You don't hit the brakes. You're not necessarily going up or down the hill. You're just going down the road, and eventually, you stop moving, right? So the question is, what happened to all that kinetic energy, right? You had kinetic energy. You were moving. Energy can't be created or destroyed. So then what happened to the energy? So the answer is, boom, this one, C. It was released as heat. All you need to know is that energy is converted. It's always, again, it can't be created or destroyed. It can only be converted from one form to another. So as you're slowing down, your kinetic energy has to be going somewhere. Where is it going? It's being released as heat. Because as I've said, chapter 5, when you go from one form of energy to the other, right? When you convert from one form to the other, you always lose some as heat. Obviously, it's not going to be converted back to chemical energy because, first of all, I never said anything about chemical energy in that question. It's not going to be returned to the bicycle, and it's not going to be used to chemically break down the tires. So yeah, the answer is it's released as heat. Any questions about number 6? All right. Again, obviously, if you get that question, it might not be about bicycling. But the idea here is when you change energy from one form to another, you're going to lose some as heat. That's the concept for number 6. Number 31, a cell that neither gains nor loses a net amount of water at equilibrium when it's immersed in a solution is what? Well, that's a simple question. It's a long-winded question, but the answer is isotonic, right? If it's not getting water and it's not losing water, that means it must be equal to its environment. The solution, right, whatever solutes it has, must be equal to the outside, must be equal to the environment. That way, the water has no reason to come into the cell or leave it, because everything's equal. Any questions about number 31? All right, number 9, humans convert approximately how much energy that is stored in food to useful work. That's just one of those numbers. I said I'm not big on numbers, but you need to know this one, and the answer is 34%. So in this case, yes, there's something to memorize. Humans convert 34% of their energy stored in food to useful work. And going back to that previous question we just looked at, what happens to the rest of it? For the most part, the rest of it is lost as heat, or used as heat, if you want to look at it that way. That's why we are 98.6 degrees Fahrenheit, because the rest of that food is being developed as heat. Any questions about number 9? That's a good independent work question too, now that I'm thinking about it. If you're interested in other animals, or, yeah, but why not? Like, if you like dogs or alligators, dolphins, whatever, see if the numbers are out for those animals. Anyway, The principle that energy cannot be created or destroyed is known as the conservation of energy. There's not much else to say about that. It's pretty simple. I probably won't be on the exam because it's so easy, but we'll see. Any questions about number 4? All right. 10 kilocalories are equal to how many calories? Remember, a kilocalorie, the kilo is the key word there, is 1,000 calories. So just literally you can just read that and just translate it. So 10,000 calories, right? 10,000 calories are equivalent to what? 10,000 calories. There's not much else I can say about that. Any questions about that? All right, number 7. Which of the following is a measure of randomness in a system? I told you didn't need to know this. This is important mathematically when you're really looking at chemical reactions, but we don't do that in this class. But the answer is entropy. Won't be on the exam, so let's move forward. Osmosis can be defined as what? Let's just look at all the options. Diffusion of a solute. Nope, it's already wrong, right? Because it's about water. So diffusion, you had me at diffusion, you lost me at solute. The diffusion of any small molecule. Nope, again, you had me at diffusion, lost me at small molecule. Active transport. No, you lost me at active transport because it's not, it's diffusion. Therefore, by process of elimination, we just happen, and it happens to be the last option, the diffusion of water across a selectively permeable membrane. There you go. Any questions about number 28? Number 13, ATP energizes other molecules in cells by transferring what? If you can just remember that ATP is the fully charged battery, and ATP means adenosine triphosphate. Remember that's the fully charged battery. And remember that ADP is the dead battery, right? Adenosine diphosphate. So triphosphate has 3 phosphates, diphosphate has 2. If you just remember that, then obviously you can remember the fully charged battery, so to speak, has 3 phosphates. The dead one has 2. So obviously the fully charged battery gives up a phosphate. That's how it gives up its energy. So that in mind, let's look at the options. And it's transferring what? It's transferring a phosphate group. And again, if you can just remember, obviously in the names, ATP, ATP gives up a phosphate. That's why ATP goes from ATP to ATP. So obviously it's giving up a phosphate. Any questions about number 13? All right. Number 5, a boulder at the top of a hill contains blank energy. Very simply, since this question didn't say anything about it moving, it just says it's at the top of a hill, the answer is potential, right? Potential is stored energy, whether it be because of its location or its structure, like the way a molecule is built, that's potential energy. Now, if it said, and I might reword it this way, a boulder rolling down a hill contains blank energy, then the answer would be kinetic, right? It's exhibiting kinetic energy because it's moving. And again, this might be one of those questions that's worth 2 points. If you guess potential, 2 points. If you guess kinetic, at least you chose something that is involved in that conversation. And if you guess no energy or conserved energy, you just really off base and get 0 points. Any questions about that? All right. When you take this test, I recommend having a scrap piece of paper and something to draw with, because this would help if you could draw it and see what we're looking at. A balloon that is permeable to water but not glucose contains a 10% glucose solution. So in your mind, picture a balloon that has sucrose solution in it. It's 10% sucrose, but it's 10% glucose, right? The rest of it's water, whatever, it doesn't matter. The point is it's 10%. And now, this thing will not let glucose in and out. It will let water in and out, but not glucose. So you take that balloon and you put it in a beaker that has a 5% glucose solution, right? So it's more concentrated inside the balloon than outside the balloon. Which statement correctly classifies the solution? So first of all, we just know, as far as vocabulary is concerned, 10 versus 5, right? So it's a higher concentration inside than outside. So we could say that the balloon is hypertonic to the beaker. The same way, or the same thing we could say in a different way is the beaker is hypotonic to the balloon, right? So let's look at the options. The solution in the beaker is hypertonic. No, because 5 is lower than 10, so that's not it. C, the solution in the balloon is hypotonic. No, because 10 is greater than 5. The solution in the balloon is hypertonic. Yes, relative to the solution in the beaker. There we go. D is the answer. because 10 is greater than 5, right? The balloon is hypertonic to the beaker. Now, in this question, if I were to give you this question, I might not use glucose. I might say sucrose or salt. It doesn't matter. The glucose part does not matter. I also might change the numbers. So it might not be 10% and 5%. It might be 50% and 2%. Who knows? Also, the balloon might be less than the beaker in the question on the exam. So I might flip-flop them. And this, again, assuming I didn't change this at all, I could change this to say the solution in the beaker is hypotonic relative to the solution in the balloon. You guys see what I did there, right? I said the same thing. I just used different words to say the same thing. So when you're studying this question, do not memorize that exact answer. Just understand that hypotonic means that there's a lower concentration. And hypertonic means that there's a greater concentration. Any questions about number 29? All right, number 27. That'll be the next attendance word, if you will, or I guess the number. Will just be 27. You just write the numbers 27. You don't even have to spell it out. 27 is the next attendance word, so to speak. Anyway, facilitated diffusion across a biological membrane requires what? And moves a substance blank against its concentration gradient. So first of all, if it's facilitated diffusion, first of all, it's diffusion, right? So we know it doesn't need energy because diffusion is passive transport. So we know it doesn't need energy. We know if it's diffusion, it's going from an area of high concentration to low concentration. That's the things we know about diffusion. Then add the word facilitated. So the fact that it's facilitated means that it's still just diffusion, except it needs the protein to allow it to happen, right? It needs the doorway, so to speak. It needs something to allow those things to come in and out. So, transport proteins. Yes, it does require a transport protein down. But just so happens that, yeah, that's the correct answer. Facilitated diffusion requires a transport protein, and the substances are still going down the concentration gradient because it is facilitated diffusion. B is incorrect, or excuse me, C. This one's incorrect because it's still facilitated diffusion. It doesn't require energy. Therefore, it's not going against the concentration gradient. It's going down the concentration gradient. This one's almost correct because it starts off with proteins, transport proteins. That part's correct, but then it says against the concentration gradient, which is incorrect. And this one's starts off incorrect because it says it requires energy. That part's incorrect, but then the second part is correct because it is going down the concentration gradient. So, in this situation, if you were to get this question on the exam, again, this might be worth 2 points. 2 points for the exact correct answer, which would be transport proteins and down. And then you might get 1 point for guessing these, because at least those two are half correct. And if you were to guess C, you would get 0 points because it is completely incorrect. Any questions about that? Okay. Speaking of which, on exam 1, some of these weren't so half correct and correct. Some of them were just straightforward, like here's the correct answer, here's a bunch of incorrect answers. But on those, I might have to add some. You must have remembered some of them had really, really stupid off-the-wall answers that made absolutely no sense. They were clearly there to be funny. And if you guessed those, you got 0 points. And if you guessed anything else, you got at least half a point. And some people still got 0 points, which tells me, I mean, you must not be paying attention to what you're answering. So please pay attention to the exams. Anyway, any questions about 27? All right, number 34, in a hypotonic solution. So again, picture this, the plant cell is in a solution. The solution is hypotonic, which means it's a lower concentration than what's in the cell. All right, it's a lower concentration than what's in the cell. Therefore, I know water wants to go into the cell. All right, now that I have that in my mind, let me read the question. In a hypotonic solution, the plant cell will what? Burst would be about half correct. So if you guess that, you might get partial credit because the fact that it's in a hypotonic solution means water is coming in, right? So burst, at least you're on the right track. The correct answer is becomes turgid because that means the water's coming in and it's basically just stiffened up. Yeah, so that would be full credit. Burst, you probably get half credit, and these other would be 0 credit, 0 points. Any questions about number 34? All right.
Is chapter 6 and chapter 7 study guide required? Like we have to do?
No, none of the study guides are required. They're all,
And it's only 2 points?
yeah, 2 points to do them for independent work. But the most important thing here is that those are the questions that are going to be on the exam. So hopefully you do it just for the sake of preparing for the exam.
Sure.
Same with this one, chapter 5, same situation. Which process could result in the net movement of a substance into a cell if the substances are more concentrated in the cell than the surroundings? So again, it might help you to draw that out if you need to. But we're talking about stuff, getting something to go in the cell when there's already a bunch of stuff in it, right? So this is the opposite of diffusion. We're going up the concentration gradient. We're going from low concentration to high concentration, and that's going to require energy, which means active transport is the correct answer. Any questions about that? We only have 10 minutes. I doubt we'll be able to finish. So before I keep going, I'll remind you, everything I'm doing right now, I can do with you one-on-one. So if you want to meet me in office hours or schedule time to meet me outside of office hours to go over chapters 6 and 7, we can do this one-on-one, and I can go through each question with you. But moving forward, which statement about an enzyme is true? All right, an enzyme becomes another enzyme after a reaction. No, enzymes don't just all of a sudden change to a different type of enzyme. No. An enzyme is used up in a chemical reaction. No, that's the opposite, right? An enzyme is based on, one of the things about enzymes is they're not used up, right? They facilitate the reaction. They aren't used up. An enzyme works on a broad range of substrates. Incorrect. The part that makes that incorrect is that word broad range, right? They're very specific to what they're working on. So obviously, by a process of elimination, an enzyme's function depends on its three-dimensional shape. So that's the correct answer. For this question, I would probably reword this to make this the incorrect option. So I would probably reword it to say an enzyme's function depends on its molecular weight, right? Which would therefore make that incorrect. And I might make this one say an enzyme works on specific range of substrates, in which case that would be correct. Or I might say an enzyme is not used up in a chemical reaction, in which case that would be the correct one. Or I might say an enzyme becomes ready to be used again after a reaction, in which case that would be the correct answer. So any questions about number 23? Number 15 is pretty straightforward. Enzymes are usually what? Proteins. As far as we're concerned, all enzymes are proteins. That's all we've ever talked about is proteins as enzymes. If you want to for independent work, you can look up what the exceptions are, but you don't need to know it for the exam. Enzymes are proteins, and proteins the proteins are based on the three-dimensional shape. Any questions on number 15? Number 42, which of these molecules spontaneously form membranes when mixed in water? Oh, skip that. We're not gonna go over that. Not gonna be on the exam. Excuse me. An enzyme's function is most dependent on its what? We just said that, its shape, right? There's not much else to say about that. Here's one of those where it will really help you to draw this out. On a lab bench, there are 4 balloons filled with identical volumes of different salt solutions. So balloon A has no salt in it. Balloon B has a little bit more salt in it. Balloon C has even more salt in it. And balloon D has the most salt in it. There are also 4 beakers filled with identical volumes of a 10% solution. The balloons are semi-permeable. That is, only water can enter and exit the balloon. All 4 balloons are placed in separate beakers at the same time and are allowed to come to equilibrium. At equilibrium, which is the correct order of balloons. All right. So, again, this would be easier if this was drawn out. I'm trying to picture this. Balloon A is 0% and it's in a 10% solution, right? So the water is going to want to leave the balloon and go to the solution that's in the beaker, right? Because it wants to dilute it, right? So we know balloon A is going to get really small because that's a huge difference between 0 and 10%. Balloon A is going to lose a lot of water. Balloon B, again, it's 5% and it's in a 10% solution. So it's also going to lose water because it wants to dilute the solution. But it's going to lose a little bit less water, right? Balloon A is going to be really, really small. It's going to lose a lot of water. Balloon B is going to lose water, but not as much because there's less of a difference between 5 and 10 and 0 and 10. Yeah. Did I say that right? Yes. Balloon C is 10%. Whoops. But then again, so is the solution that it's in. So balloon C isn't going to get smaller or larger because it's isotonic, right? It's in the same solution. It's not going to change shape. Balloon D is in 15, or is 15. Come on. Balloon D is 15% solution and it's in a 10% solution. Therefore, for balloon D, the water is going to go into the balloon, right? Because we want to dilute it. So again, balloon A is going to be really, really small. Balloon B is going to be not as small as that. Balloon C is not even going to change shape, and balloon D is going to get bigger. So that being said, from largest to smallest. So there we go. So it's going to go D, C, B, A. That's this. There you go. There's the correct answer. D, C, B, A. When you get that question on the exam, because you will, obviously it might not be salt solution, it might be sucrose solution, it might be glucose solution. Who cares? That part really doesn't matter. The numbers might be different. So balloon A might not be 0%, right? I might change the numbers. The question, all right, so it might say which of the correct order is the balloon from smallest to largest instead of from largest to smallest. So again, the idea here is understand and study the concept. Don't memorize the correct answer verbatim. And the next word for attendance is this word that I've highlighted, and then I'm swirling around right there. One that's highlighted, not that whole thing, but that one. This one right here is the next word for attendance. So any questions about number 41? So again, it would help to have scrap paper and something to draw with. Usually that would help me to be able to visualize that. Another thing is, because this is the time exam, I might shorten that question. So I'm probably not going to say it's on a lab bench and say all that stuff. I probably won't say the balloons are semi-permeable only to water. So that stuff is just given, right? So if you get that question and I don't mention the fact that the balloons are semi-permeable, don't think this is a trick question. Nothing's going to happen because they're not semi-permeable. Don't worry about that. If I leave out those details, that's all to keep the question shorter so you don't spend valuable time reading it. Number 2, energy can be defined as what? The answer is simply the capacity to cause change, right? So A is the correct answer. It's just not the most correct because yes, energy can cause movement. Calories are a measure of energy as opposed to energy as a measure of calories. And it's definitely not a measure of disorder because that's entropy. So the answer is the capacity to cause change. That would get you full credit. The capacity to cause movement would probably get you partial credit because at least that's correct. It's just not the most correct option. Any questions about number 2? 38, I'm going to completely skip. I would talk about it, but we're short on time and it won't be on the exam. The region of an enzyme to which a substrate binds is simply called the active site. There's not much else to say about that, right? You have the enzyme, you have the thing that works on that place where they come together. It's called the active site. The combination of sucrose, sucrase, and water. This looks familiar. We've already had this portion of it. Glucose, fructose, which component is the enzyme? We already said it. If it's on both sides of the reaction, it's an enzyme. If it ends with ASE, it's an enzyme. So the answer here is sucrase. That would get you full credit. Maybe I'd get you partial credit for sucrose because at least you phonetically were in the right ballpark. And again, when you get that question, I'm probably not going to use those exact substances, but the idea here is you need to know enzymes are not used up in the reaction. Enzymes end with an ASE. Any questions about number 20? Number 37, when a person cries. That's exocytosis. We're not going to talk about that. That's not going to be on the exam. The sum total of all the chemical reactions that occur in an organism. I just said that once. The answer is metabolism. You just need to know it. 36, active transport. It's active, right? So can involve the transport of ions. That is true. Move solute with a concentration gradient? No, that's just diffusion. Does not use ATP? No, it needs ATP. Is a type of facilitated diffusion? No, active transport is the opposite of diffusion. So the answer is can involve the transport of ions. And I probably didn't even talk about that very much in the lecture, but I did say you do know that active transport goes against the concentration gradient. So you should know that B is wrong, right? Even though you might not know C is correct, you should know B is wrong. You should know ATP is wrong, and you should know D is wrong. And also for the exam, I probably would change it. So I might say cannot involve the transport of ions, which would make that an incorrect answer. And I might change this to say move solute against the concentration gradient, in which case that would be correct. Or I might say does use ATP as an energy source, in which case that would be correct. Or I might say is not a type of facilitated diffusion, in which case that would be correct. Any questions about number 36? Number 18, which statement about enzymes is true? Enzymes are the reactants. Nope, they're not the products or the reactants necessarily because they're on both sides. So A and B, or this one's wrong, this one's wrong. Enzymes catalyze specific reactions. That is correct. Enzymes functions require inhibitors. That is incorrect. Sometimes inhibitors do act on enzymes, but they're not required. So the answer is C. And that's where we're at. Like I said, we didn't finish it. We didn't even finish chapter 5. So if you have questions, let me know. You have a full week, a week and 2 weekends to study for this exam, and I'm here to help you. So that's it. Let me say the last word for attendance is going to be beaker. Beaker is the last word for attendance. I'm going to be online for office hours. I will not be physically in my office. You guys have a great weekend. And that's it.
So when I looked at the lab for next week, is it just like cropping the
Yeah.
photos?
The concepts are really simple. It's
Right.
just, I found that most people just have problems like just figuring out how to crop stuff. So that's what when we meet on lab on Wednesday, that's basically what I'll be doing is showing people how to do it. Actually, I think there's even videos embedded in the lab that show you how to do it.
Yeah, I saw the link.
You said it was Beaker, right?
Yes, speaker is the last word for attendance.
Just didn't get time to click on the videos, but I saw that there were videos on there.
Gotcha. Okay.
So,
Have a good one.
me too.